From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 67, recorded on April 21, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores. What does it explore? I forgot. Oh, it's the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Great to be back. Hello, everyone. And great to be live streaming again. That's fine, Vincent. Yeah, I think we do explore. We explore the world around us, the diversity around us, try to make some sense of it. Obviously, recently we've been very concerned about the diversity of one biological entity, SARS-2, and we'll pick up on kind of some updates in that space today.
Yeah, I was going on TWiM. I say the podcast that explores unseen life on Earth. That's what I was doing. I'm just not a good anchor, but you get what you get. So we got a bunch of people here. We have 26 people already. That's cool. We have
Nice.
Andrew from New Zealand. That's cool.
Good to see you again, Andrew.
We have Plymouth, UK. Look at that, Coast Walker. And Sean
Fantastic.
really likes your beard, man.
Thanks, Sean. Yeah, so been letting it roll here in the pandemic. I did, Vincent, just get my second vaccine dose on Friday.
Very good.
Yeah, and I'm going to bring this back to the beard somehow. But so the game plan is once we're at that 2-week, kind of 3-week post second vaccine mark, my game plan is to get out the old electric razor and take this thing down a few notches, come out of hibernation, come out of facial hair hibernation in a sense.
What's the story in your state now? How are cases going?
Yeah, so I think it's coming down a fair amount, which is good news. And the vaccines are going up. And so I think I just saw yesterday, first doses now are approaching 1 million folks, which doesn't seem big but if your state is only several million, 3 million, that's actually starting to make a dent. And with second vaccinations also picking up steam as well. And so it's been open for a while. And, if anything, we're probably approaching that tipping point where supply will start to continue to sustain or increase, but demand might go down as people are more hesitant or the other issues around some of that. And so we'll see. But at the moment, very few deaths in the last couple of weeks. And cases, sometimes kind of sputtering and going up and down, but the overall trend lines are looking favorable. Good. How about there in New York? What's the latest kind of situation?
Well, we still have cases, but deaths are way down, right? And that's good. So, and the problem is that, as Daniel Griffin says, a lot of young kids are now in the hospital. That's who's getting infected, right? So that's not great, but the percentage of serious illness is lower anyway. So I think, we've got vaccine into a lot of people, and we're doing great. 3 million doses a day, I think. It's really cool.
Yeah, that's pretty impressive. I hope if that can sustain for a while, that would be fantastic. Yeah.
Auden. I think Auden is the way you say it. Like Audi, Auden. Super chat. Thank you so much from Oslo, Norway. Hey, look.
Fantastic.
Randy
Skull.
From Trinidad.
Great to have you along, Randy. Thanks for joining.
Mic level is low, huh? All right, I'll crank it up. I'll crank mine up. They're saying mine is low. You're going to crank it up and don't complain if it's too loud, folks. Is that better?
I'm glad I'm coming through. Usually it's the other way around. So
well, I'm a soft-spoken guy, Wasn't that a ZZ Top song with the guys with the big beards? That ought to be you, right, Nels?
Heading that way. I might have to get on the TikTok or something to really try to capitalize here on this is probably the longest I've gone without kind of any facial hair related grooming. So kind of enjoying the ride, to be honest, but who knows? Stay tuned. Maybe next episode, TWiEVO 68 will be.
Yeah, that'll be next month. So you're going to have your second dose by then. So we're all set.
Second dose in, and the spike proteins are, I would imagine, in this transient transfection of a vaccine, that the spike proteins are being made even as we speak, and my immune system is responding. It did respond over the weekend, I'll say. I was in pretty rough shape, tired, achy, chills, everything like that. I resisted the urge to take any NSAIDs, any Advil. They said to take Tylenol. I just decided to ride it out and kind of sleep it off, but feeling a lot better and quite optimistic, actually, as well.
Very good. Well, we have a paper for you today. Nels, want to jump into this?
Yeah, let's do this. So I think I saw we have someone from the UK along for the ride here. And so we will be considering some work out of the UK in one of those variants of concern, maybe the first one on the map, the B.1.1.7. And this is, so it's not quite a paper, it's not quite a preprint, it's a note on virological.org, a resource that we've been tapping into over the last several months on a few occasions. Here, Vincent's putting it up. Thanks for that. And so titled here, Recombinant SARS-CoV-2 Genomes Involving Lineage B.1.1.7 in the UK. And the keyword here is the first one, recombinant. And so I wanted to really spend a little bit of time thinking about recombination. We've talked about this a bit, but to kind of dive back in from an evolutionary standpoint, thinking about recombination, what it means for the diversification of viruses like SARS-2, other viruses, the diversification of us, actually. I think we'll maybe take a detour into that as the conversation unfolds here. But this is important, and I think, still a little bit underappreciated. So this idea that SARS-2 is diversifying, or has the potential, certainly, to diversify by recombination. And so this is different than kind of the classic point mutation view, where one of those RNA letters gets changed to another RNA letter, and that's the basis of a mutation. Of course, we've been tracking, including in B.1.1.7, some of these mutations at positions in the spike protein. So 501, change in the RNA letter that changes the amino acid, changes the shape of the spike protein. That's been discussed, continues to be pored over from a research standpoint. But this, and so those are those point mutations, one letter at a time change, and those can happen in flurries and somewhat quickly. But recombination is kind of a shuffling of the deck. So the idea that the virus can, as it's replicating, as it's making another copy of the RNA genome, the polymerase, the RDRP that's doing this, can actually switch tracks, in a sense, from the strand of RNA it's sitting on, from the original virus genome, to another strand. And that could be one that had already just copied very recently, in which case it might be sort of a seamless recombination event where you hop templates, and the template's the same the rest of the way. So the replicated product looks the same. Or, and this is what we'll tackle today, if there is a co-infection with a different variant, or even, how far could this go? How diverse could this be within the coronaviruses? The possibility that the virus, as it's replicating, switches templates and then picks up on one that's quite different, that's diversified. And the product of that would be almost like a genomic scramble. You start on one, you end on another. And so we'll look at that. The crew, the authors here.
Nels, that, sorry to interrupt you.
No, go right ahead.
The coronavirus polymerases are very good at skipping, because to make mRNAs, they actually skip in that process. They skip from one end of the genome to the other. And that's how they make the nested RNAs. So that is probably why they're really good at recombining during other processes as well.
Yeah, really good point, Vincent. And because we've talked about this a bit, these are, for RNA viruses, these are massive. These are the super tankers of the RNA virus world. 30,000 letters, 40,000 letters. That's a lot for an RNA virus. And as a consequence of that, proofreading activity encoded in the virus genome, so that unlike some RNA viruses with smaller genomes, fewer genes, that are a little more sloppy with that copying mechanism, this one does better. So the point mutation rate is actually down, maybe an order of magnitude, even 2 orders of magnitude from some of the more promiscuously replicating RNA viruses. I'm thinking your old friend poliovirus. I'm thinking HIV, for example. And so you could imagine that recombination kind of covers that deficit of diversification in a sense. And so that's why it's probably important. It's a little bit harder to study, a little harder to diagnose. It's not quite as quick, and we are still kind of figuring out how to calibrate to measure and understand recombination. So I think that kind of lags in our understanding.
So let me just interject something, since you mentioned poliovirus there. With poliovirus, the virus exists very close to the error threshold. And if you add a mutagen to an infected culture, you only need to increase mutation frequency by 2 or 3-fold, and the virus is extinct. If you can also introduce a change in the polymerase to reduce recombination, because that's been known that David Barton's group found this one amino acid change will reduce the recombination frequency. And then in that background, if you add the mutagen, it's even worse. It can't tolerate any increased mutation, which shows you that recombination is essential for rescue from error catastrophe.
I love that. Yeah, that's really cool. Great illustration of the point. Yeah. So, okay, so let's back up. So who did this? Who wrote this note? So it's some familiar faces. So Andrew Rambo, who I think helps run this virological.org, Oliver Pybus, and then a couple others. One, the last author here caught my attention. So I'm familiar with Nick Lohmann's work. So he's, first of all, he has a spectacular Twitter handle. It's worth it just to consider some of the work based on his Twitter. Pathogenomanic is his handle there, combining the pathogen genome with his actual name. And he's run his crew at the University of Birmingham in the UK for, I think, more than a decade now. And this doesn't come up in this note, but I just wanted to mention it a little bit at the top. Some of the work that Nick is really well known for is really developing some of this technology of long read sequencing. So, and in particular, the Oxford Nanopore platform, which is really, I think, in cool ways brought the ability to do genome scale sequencing just into the individual lab, right? So these are these devices. The MinION is probably one of the workhorses of Oxford Nanopore. It's like the size of a harmonica. You can plug it, has a USB plug into your laptop, and you can squirt a little of your genome library in there and run this now, in 24 hours or less, and, on your laptop start doing data analysis. So anyway, Nick Lohmann has had a really big hand in bringing this, especially into kind of infectious microbe space. So I think
Would that be this, Nels?
Yes. Yeah, that's right. That's great. So that's exactly it. The MinION from Oxford Nanopore shown plugged into a laptop there. You can, if you just go outside my office here, you can see that some of those, similar images there. We've been doing this with a different virus, the pox virus is vaccinia virus, the vaccine strain, and which is an even larger genome, 200 kilobases. We kind of took a pause on that, obviously, for a few months. But yeah, using this exact same technology. Nick really advanced it in bacteria. So E. coli, pathogenic E. coli strains. I think the longest read, this is a while ago, and I'm just kind of foggy on my memory exactly, but it was like a megabase. So imagine you're using this instrument to thread, in this case, DNA from E. coli through this little nanopore, this little pore, and detecting the electrical disturbances as it goes through to get up to a megabase of in a single run, in a single read. And so this is really cool for some, has some challenges and some drawbacks, but it's rapidly improving, in part because of the advocacy and energy of Nick Lohmann and others. And so in addition to bacteria, he actually moved a little bit into real-time surveillance of Ebola virus, one of the recent outbreaks in Africa, to, because you have that little small device that you were just showing, you can bring it into the field. You can bring it to, rural clinics, and again, set up shop and return basically real-time genome analysis from samples collected from patients. And so some really exciting advances there. He also worked, I think, with Zika virus in Brazil as well. So yeah, that's right, Andrew. Yeah, it has this sort of Star Trek tricorder energy to it. And in fact, they even have some smaller devices now that you can hook onto your iPhone. What's that one called? The Smidgiant, I think it's called. And so it's going even farther. So pretty soon that'll just be, like probably an iPhone 20. You'll have that option at Apple's webpage. Do you want a DNA sequencer, an RNA sequencer as part of your, for an extra $50 or something?
Are these reusable, or do you have to throw it out each time?
Yeah. So the yes and no. So the flow cell that goes inside of that device is a consumable. And that's where that's sort of the business model of Oxford Nanopore, right? Is they want to sell you. They might even give you that sort of exterior shell, and then they want to sell you flow cells. There are some tricks though. You can wash them and reuse them a couple of times and really push that, depending on it. So if you're doing a coronavirus, for example, if you're doing SARS-2 in an Oxford Nanopore device, you can multiplex. So you could say, okay, because the genome is 30,000, which we're saying is big for a virus, that's small for a sequencer. And so you might do a dozen at a time, barcode your samples, and then use the output to then sort it all out. So you kind of mix them all together and then computationally isolate them at the end. So really, really fun technology. So I've been kind of starting to get excited about this. That's not at play here, it turns out, in this note, but actually the long reads are really useful. Excuse me. And I wouldn't be surprised if the Lohmann lab is continuing to follow up, and we might keep our eyes open to see if some of these recombination events proposed can be confirmed by this long read sequencing. Yeah, thanks, Molly. So guilty as charged. It's been a few years. I'm a native Minnesotan, Twin Cities accent. Actually, I'm kind of glad this came up, so that obviously as a nation, in the last 24 hours, all of our eyes have been, I think, at least from time to time here, on the Twin Cities, and what was happening in the trial of George Floyd's death. Here, just personally, kind of taking a detour, obviously, from our main conversation, but what a relief that we had a measure of accountability for that really pivotal event. This is where I grew up, actually, Vincent. So the neighborhoods, my family, my neighbors, my friends, and really kind of keeping a close eye on this, and really feeling not quite celebration, because it's such a sad outcome,
that's
such a
sad occasion. To celebrate,
but a little bit hopeful that at least there is some accountability on that, towards justice a little bit, and hoping that we keep moving in these positive directions somehow. So back to this. So and Remo with a great question here, how the virus phylogenetic tree is built. This is actually an important, not just a detail. This is really important. I'll tell you what, why don't we'll chase after a little, or explain, put on the table some of the findings here, and then return to that question, because I think it's really important. What it actually does, it influences the recombination, can, again, not only shuffle the virus genome, but then when it does that, almost necessarily the relationships between viruses also become scrambled. That phylogenetic tree changes a little bit. And so their virus trees are built the same way that we build it for, say, primates or mammals or other species, in this case, based on the RNA sequence, the relatedness to it. But recombination can really influence, and we can use actually some of the differences in phylogenetic signals. Some of the detectors of recombination actually do this. They draw many phylogenetic trees, ask where are they a little bit different or incongruent from each other, and then propose recombination breakpoints that might explain that scrambling of the branches of the tree. Okay, so but here, the authors, I took advantage of a massive data set that was emerging around the time, around the turn of the year, from 2020 to 2021, and this is the emergence of B117. Talked about a lot, obviously, and I think actually we might return to this at the end. I was just looking at covariance.org, which tracks the frequency of some of the variants of concern, of interest, of the major variants. And in the UK, for example, numbers, again, thankfully coming down quite a bit. But B117 of the sampling that's being done is something like 97% now. And that's kind of plateaued. It's been near that 100% for several months. It was actually in figure 2 of this virological note. They show this as early as end of February, into the frequency around 90% or so. Yeah, there it is. So it's that big brown part of the graph there is B117. And then it doesn't show it here because this is just frequency. It's not total numbers, but especially around the turn of the year. And that would be a little bit to the left of the middle of the graph, where you have that lighter brown, that beige, is another B lineage, sort of a similar name, B.1.177. There's an extra one in there. Close relative, but not exact. And so the interesting thing is, as the authors were sort of trolling through some of the genomic sampling, the genomic surveillance, they noted that some of the samples, if you look at them, they look mostly like B117, but lack some of the defining mutations. And so this is there's 20 or so mutations that sort of define the lineage. And it wasn't just sort of like randomly missing them. It's like there would be a block missing. Like maybe you have the first 10 defining mutations, and then the second 10 are missing from as you kind of read from left to right through the 30,000 letters or so. So, a couple ideas, what could be going on here. But when they kind of started piecing it together, they noticed that actually if you take, for example, the second half of B.1.177, it was, this back end was basically replaced with some of those mutations. And so that is pretty suspicious. You have recombination, that you have sort of chunks of defined mutations coming together for the first time. And so, what's going on here? So taking a closer look, actually, can you put up, Vincent, figure 1? I think this really kind of nicely, this is great. So this sort of illustrates what I'm saying. So blue here, so this is, we're looking now at the first 4 are actually 4 groups, and that will become an important detail. So each of those horizontal lines represents more than one virus that was sampled, but it looked very similar or basically identical in terms of the variation. And then the bottom 4 are individuals. And so the blue bars there are consistent with being the B117, and we might consider this the parent, you could call it, virus at that point. So, and that's a little bit of an arbitrary definition, but then the like if we look at Group A, most of it, the first 2/3 is this beige color. That's B1.177. In terms of the mutations. There's a little, and actually it's important here, and this is a really cool point, is that gap, and there's bigger gaps, especially near the bottom. Doesn't mean that it's like deleted or that the sequence is missing. It's that you can't diagnose it, because in that region, both of these virus lineages, B117 and 1.177, have the identical RNA. And so what you're looking for, so the implication of these types of patterns, right, is that the virus was going along, and let's just take Group A at the top, and was replicating. And this was one of the, we'll just call it the beige viruses, brown viruses. And then it got kind of about 3/4 of the way through and hopped over to the blue virus, B117. And that's the recombination event. And so then it finishes there. And so it looks like this hybrid or mosaic virus. If the viruses are identical, then what that gap is called, or what you're looking for, is when did it switch? It's called the recombination break point. It's where you break off from one genome into the other. And that can be hard to define, especially in closely related viruses. And so this might get a little bit to Rima's question. When we think about the phylogenetic trees, the fact that these sit so closely is because they're nearly identical. And so in these recovered viruses, that's the prediction, actually, is that you could explain finding these kind of virus chameleons. They've switched their color, in a sense, in the second half. Or, if we look at group C, the reverse happened. It looks like, so most of the parent sequence here is mostly B117, the blue, and then just the end are some of the unique mutations different from B117 for the brown virus in that
Nels,
case.
can I ask you, so the gap, you're saying it's too close, but in some cases they can assign that same area to blue or beige. So what's that about?
Yeah, good question. So what's happening there is, like, let's take, for example, B and let's see, what would be a good one, actually. So if we took C and let's take C and D.
Mm-hmm.
So it's kind of the reverse. So it's, kind of roughly in the 3'. That's actually right around the spike, which has some kind of functional interest there. So in that case, so the way that the mutations are accumulating, and there's probably, we know that there's 22, I think, in the blue, and let's just assume there's something like 20 quote unquote defining or unique mutations for the brown here. And so depending on how those mutations are spaced out is going to be what defines how big that gap is, or how hard it is to find another change. If you see what I mean. So so if you have two unique changes between the two, kind of evenly spaced, then at 50 RNA bases away, then that gap will be 50. If they're spaced out like 3 kb away, now all of a sudden you have a bigger gap. So it's how they're arranged, right? And so ideally these are random mutations, point mutations, and so and a few probably indels here, like very small deletions and things like that. But these are happening randomly, and so they're not just sort of evenly across. If you had perfect markers, right, then you would be able to see gaps that were identical.
Okay, got it.
Yep. But it's an important point, Vincent, that you're raising, an important question, because you want to be a little bit careful about how do you diagnose whether this is truly recombination or other events. So, and there are, and I think the authors really nicely, they actually have a section called, I think, alternate explanations from what they're proposing there, right? And I really appreciate that, because it shows, that they are thinking about this rigorously, which, and for recombination in particular, you kind of need to do. And I should say, maybe before getting into that, they're not using those long reads that I was sort of singing the praises of. They're using the kind of, so the surveillance mechanism right now is, I think there's almost 100 reactions that you do. So you get, patient comes in and you want to understand, what does the genome look like of the SARS-2 that's infecting them? You isolate the RNA, and then you set up 100 reactions to amplify across the genome these little 350 letter fragments that overlap a little bit, and then you tile it all together. And then that's what gets deposited in the database. That's the data that they're analyzing and comparing. And so, the alternate explanations, you have to be worried a little bit about just that process of, if you tile it together in a slightly, just as the algorithm is proposing, this piece should overlap with this piece down the way. If that gets scrambled a little bit, or it amplifies in a strange way, you'll get a mosaic potentially, but it won't have the same pattern. And so that's where they point out, that's not happening here. You actually have these groups, you see it multiple times. It's in independent times and coming out differently. If something like that was happening, groups A, B, C, D would all collapse most likely into just one group. It would all look the same. It would be the same artifact. And they're doing this, the samples are from different labs. They're from, happening in different locations. And so that kind of takes away that alternate explanation. However, even in addition to that, there could be other artifacts. So things like contamination in the lab, where you think you're sequencing one sample. In fact, there's a little bit of a second sample in there. You've got a little bit of brown with your blue, or blue with your brown. And just by chance, as those probes are going through, some might grab brown, some might grab blue. Another possibility is that it's not contamination necessarily in the lab. It's contamination it's natural contamination in the patient. The patient is actively co-infected by brown and blue virus. And so
Mm-hmm.
your sample you collected might be totally clean, but it might return different fragments like this, even though it hasn't physically recombined. Just the presence of the genomes there
Yeah,
and the
sure.
way you sequence it could have that property. And so they also account for that as a very unlikely explanation, given the analysis they do, the fact that they see this multiple times. So group A, for example, the fact that they saw that, I think it runs brown for a while and then it jumps over to blue, because they recovered that from several patients, that also suggests that that virus recombinant has transmitted to multiple patients as well. It happened a while ago. And in fact, in figure 2, where it shows that frequency, based on, I mean, the surveillance here in the UK over this time is, was really picking up steam. And so they can give you sort of like a date on each of the recombinants they're proposing. Those are, thank you. Those are at the top there, those arrows. You can see group A, B, C, D. And the left arrow is when, that sample was recovered. And so necessarily the recombination event being proposed would have happened earlier in time along the way. And so that's happening there as well. The final kind of major alternate explanation is, well, viruses mutate a lot. And so maybe you just happen to have this pattern of mutations that's consistent with being that kind of lineage defining for a while. And then you either revert some of the mutations or change them so it matches the other virus lineage. There, and you can't, that's like formally possible, but it's sort of a probability situation. The chance of hitting on the exact same mutations in that exact combo would take, at least probably a dozen evolutionary events. Whereas by just a simple single template switch or hop, you could, in one event, recapitulate or build that genome with those kind of unique set of mutations. And so that's an argument of parsimony. The simplest explanation is probably the one that happened. And so complicated things do happen and you violate parsimony all the time. But here, given the time frame and the other details here, that would be pretty unlikely. So there it is, probably, I think, pretty actually strong evidence that recombination is happening in real time as these variants are starting to come to a high enough level in the population that people are getting multiply exposed, multiply infected. And then the output of this are some of these viruses that have scrambled the deck, so to speak, genetically, have sort of changed their chameleon stripes.
So Nels, what do you think is the frequency of so this requires that one person is co-infected with two different enough viruses to be scored in this recombination assay, right?
Yes.
How frequent do you think it is, or do you think it's just that there are a few people and then those recombinants spread? Because with HIV, the main group of HIV diversifies into subtypes, right? Very much like these variants, which spread all over the world. And then you get recombinant forms as well, as subtypes co-infect people, especially high risk sex workers, for example, who have many clients and they get infected over and over. So what do you think is the frequency here? And you think it's really common or very rare?
Yeah, it's a great question. And I think it's one of those, great questions with a complicated answer, which is it depends. Right. So I don't, and it might be that some of the epidemiologists would be close to starting to calculate, maybe based on, there's enough surveillance now based on some of those numbers. I think the trick is there's just so many variables at play here, right? So how much virus is circulating in the population, how much diverse virus, where the lineages have differentiated from each other, are circulating. This gets back to that phylogenetic tree situation. All of the guys that are basically the genomes that are identical to each other. Well, so if you just were, because our detection is limited by having enough differentiation, we'll probably, by this rate or by these methods of detection or estimation, underestimate how much recombination is going on. The more virus circulating, the more the chances of a co-infection. I think this will be, you were mentioning Daniel Griffith's great line about human behavior of concern, right? That'll play into this as well. How many collisions are happening between infected people in real time? all of this stuff again. So I would propose that, I think there is some work in the, not a lot that I know of, but some work in this direction, is to go back to some of our core fundamental virology. So can we set up an experimental system to try to model what might be happening? I mean, so again, there's some compromises. You're simplifying your, or making assumptions about putting together experimentally different viruses and cells, probably instead of humans. And so you're pulling away a lot of that complexity, but at least you can get to a sort of biological rate of recombination. And then I think once you put human behavior or host behavior on top of that, then it gets a lot more complicated really quickly. Yeah. Yep.
So I think it's important to point out that this has been happening throughout the outbreak. It's just we're now able to see it because previously there were not so many changes across the whole genome, right? And now, as the virus expanded into many, many more people at the end of last year, basically, it diversified more. And now we can pick it up. And someone here asks, for example, is the same thing happening in India? Sure, absolutely. If you looked for it right now, I bet you would find it there.
Yeah, I think that's exactly right. And so to talk about recombination too, to step back a little bit. So recombination is a diversifying force. There's, when we talk about this scrambling or, quickly putting together new mutations, that can diversifying, yes, but not necessarily dangerous actually. So it's a mutational process like any other, in the sense that it's over, there's no kind of nefarious or evil purpose here. It's just diversifying. Most of these events will be actually deleterious. That's what we've learned from a lot of these random events, right? And in a lot of different settings is that you mostly make junk as you're randomly putting stuff together. And actually those are harder to detect, right? Because they very quickly just fall to the wayside. They don't go on. And then most of them are actually probably neutral. So the ones that are being measured here, I wouldn't be surprised if the outcome was that the virus basically replicated about the same. The question is, when we look at that, the frequencies, that it does become more complicated here, right? Because so what do we describe as B117? Does it need to have all 20 of the defining mutations? If it has the first 10, is that enough to put it in that category? Or where does it fit now into the tree? And so this is right. This is exactly Rima's question, which is a great one. We don't know. And actually, I don't know that we have the intuition or all of the tools to always. So what you call the parent virus, you could do based on, the sort of what are the mutations that dominate it, or so is I guess, is it 50% or more, or is it the most mutations? That's one way, or you might just take it away from the tree, and so that might be the easiest thing, is to just discard. You don't categorize it at all or put it into your analysis because it's sort of confusing. And so it's really interesting because then, honestly, what really matters is the function, right? Is this virus, if let's say there's a mutation in the spike that somehow it gives it a minor advantage somehow in terms of recognizing the host receptor or something like that. But if that's one mutation, but the other 19 are the parent virus of another lineage, now it kind of fits the genotype of the other tree, but it's probably acting more like one in this other lineage or other clade, other group in the tree. And so that gets a little bit interesting and complicated as well.
Well, Nels, the problem is we don't really know what most of these changes do outside of the ones that impact antibody binding, right? All the others. And in fact, the only ones that are getting any attention are the ones in the spike gene. And there are many others outside of that could have functional consequences. So I think for now you have to make the trees on the basic sequence and follow, clades. And you have to talk about a B1117 lineage, right? Not a particular virus, because they're different. As they pass from person to person, they diversify further. So.
Yeah, no, it's true. It makes life of the evolutionist a little more complicated, though, right? Because you could draw that tree based on, and mostly we do this on RDRP.
Yeah.
And for good reason, that's pretty well conserved. You need a working polymerase to be a functioning virus. And so, but recombination, as we saw, even in that sort of small set, recombination can happen over the coding region of RDRP, and so that will that'll change that relationship, and as you draw those phylogenetic trees, and so yeah, it becomes a little more interesting. And then in the rare cases, you could imagine where recombination is beneficial to the virus, and so a mutation that might give it some advantage, let's say that's not in the spike, but in encoding one of the genes that are sort of important, like maybe the replication goes a little bit faster because of a mutation in RDRP or something like that. And so it just barely outcompetes the others. And then that's brought together through recombination with a mutation in the spike. You could imagine that bringing those two mutations together for the first time through recombination. And in fact, I think, that's an open hypothesis in the lineage that has been emerging from India. And that one, really concerning when we look at the numbers there. But again, a variant of concern, human behavior of concern, all of the above, all of this complicated epidemiology playing out in real time, unfortunately, with some ongoing really tough consequences there. Ooh, interesting. So Andrew is asking, is recombination a way for the fossil viral RNA or DNA to have gotten into the human genome? And so this raises this really cool, I don't think, actually this was controversial. Just trying to bring it to the SARS-2, whether or not coronaviruses might be integrating into the human genome. And that study in particular, I think falls into one of those, alternate explanations where that was a sequencing artifact. As one of the really common things is that you get these chimeric sequences. In this case, it was like you're reading human genome and all of a sudden it's coronavirus genome and then you're back to human genome. There are, though, as Andrew is pointing out, some real events like this. So we have in our genomes things like the distantly related to HIV, endogenous retroviruses. That's not recombination, it's reverse transcription in the case of those ones, where it's getting the mechanism of the replication cycle of retroviruses includes integrating into the host genome. And then this can go on and become fossilized, basically, in these endogenous retroviruses. In fact, a pretty massive portion of our genomes are now dedicated to these fossilized virus events, the endogenous retroviruses, with some interesting consequences in some cases. But I would say in most cases, probably not recombination. There are, so certainly it would be non-homologous because you don't have that template. And there are some other interesting mechanisms at play involving retrotransposons. So it's, I think being on the lookout for what are the genetic entities that enzymatically can copy and can do sort of the dynamics here, can copy and paste nucleic acids, whether it's RT, reverse transcription from RNA back to DNA, and other steps like that. And so those are sort of the elements, and there are the potential actually for things like retrotransposons as they copy and paste their own RNA back into genomes to pick up other templates. So other messages, other transcripts. If that was a virus, you could imagine, if that was happening in a virus-infected cell, you could imagine that could be a mechanism as well. And so it looks like recombination in the sense that you have these large chunks becoming chimeric, but the actual mechanism could be a number of different things in those cases. It's a really good question. And actually, this brings me, Vincent, to a point I really wanted to make on today's show, which is about so we do recombination, right? So and this is probably a really important mechanism. And so it actually, in our own species and other animals, it raises this interesting kind of puzzle, which is, so I'll return to recombination as the answer, but the puzzle is, why does sexual reproduction exist? So if we just think about getting genes to the next generation, sort of a simple Darwinian idea, sexual reproduction is this massive compromise. So you have 100% of your genes into a gamete, the sperm or the egg, and then you're saying, you know what, I'm going to give up 50% of them to my partner. So, right. And so we emerge, for example, ourselves with half of our mother's genes, half of our father's genes. And so that's like giving up half of your genetic material is a huge compromise. Why wouldn't you just clone? Why wouldn't you just bud a second Vincent Racchianello out of your elbow or something like that? Like, the way that budding yeast does. That would be great. You'd get 100% of your genes onto your offspring there. And the reason we think there's all of this, interesting theory and ideas built the original Red Queen hypothesis was sort of founded on this idea that the reason we put up with reproduction of giving away half of our genes to the next generation is to allow the process of recombination to happen. So, when we emerge with half our mother's genes, half our father's genes, it's not sort of like segregated perfect half. Homologous recombination is happening through the process of meiosis, through the process of development, and you end up with a new combination of alleles that have sort of never been seen before. So, and why would that level of diversification kind of be beneficial or sort of outcompete the cloners, the budders who are doing 100%? We think it's because of the viruses, the things like SARS-2, the other infectious microbes that are coming at us and causing real detrimental outcomes. And so that also, like this diversification can work for the hosts too. And so that's really the big idea here, is that this diversification, it's not, and I think that's a general theme actually for all of our interactions with infectious microbes, with viruses, bacteria, fungi, others, is that this process of diversification, finding that balance point for both hosts and parasites.
I want to thank Robert from Sweden for his super chat. Really appreciate it, Robert. And here is another question from Rima. Difference between clade, subtype, strain, variant, serotype.
Yeah, Rima. Yes, you're you're hitting on the right questions here. And that is, that's for, I think, SARS in particular, for coronavirus in particular, has been both historically, so what we call a species of virus is really tricky, a lineage of virus, a clade, a family, even what we call the viruses, right? So in some of the, so calling a variant by its lineage, B117, is actually referring to this, not just one thing, but this. So there's the defining mutations, but then there's additional mutations. There's been other, and those will, and if you draw the phylogenetic tree, you can see that they associate in one branched area, and then you'd have another clade, another lineage in another area. That's true of B117 versus B1.177, which was the recombination event charted here. But then there are other classifications coming up as well, which is trying to get away a little bit from the phylogenetics. And as I was mentioning a few minutes back, thinking more about what is the key mutation here that might change the spike in a way that changes the virology a little bit. And so there, so some of the classes are then described as 501, and that would include B117, but then in that mutation in the 501 position of the spike gene, in this case, might arise independently, or you could have these recombination events that bring it together in different ways. And so that's actually to try to get away from some of the confusion that is in your question. What is the difference between a clade, a subtype, a strain? It might even add more confusion, but it's to basically center the naming of it on what is thought to be the key functional mutation. So another one is at the 484 position, which has sort of come up as well in some countries recently, or in some in different countries and at different times and in different ways. And so I don't know, it's a compromise, like how you pick to classify. And I think the outcome, no matter what, is that it's pretty confusing, even for sort of the aficionados to just look at one set of analysis and really understand where this virus is coming from. It's worth it to, if you're really interested, to kind of do a deeper dive. I think it's worth it to spend a little time at this website, nextstrain.org, that we've talked about in some past episodes. It's not totally simple, but if you're willing to give it a little bit of your patience, maybe an hour or so playing around with the controls, the graphical interface is easy enough that you can kind of start to move things around or put labels on and off based on different features, and kind of see how those trees build or how the arrangements unfold. It's actually pretty, I think it's a good exercise to get a sense of where some, but it gets complicated in a hurry. So you really, I think the more you can kind of try to simplify your data set that you're considering, so don't put all, hundreds of thousands of sequences and try to see that from like 50,000 foot view or something. But if you just pick a small set of several different, like 2 lineages or something like that, and then just sort of play around with how they're related to each other, that might be useful.
So Rima, I made a video on variants, strains, clades, blah, blah, blah here on YouTube. So there's a link. You can check that out if you'd like to hear more of us talking.
Cool.
Here's an interesting one. What happened in the mink farm? Selection pressure or recombination?
Yeah, great question. So I haven't been too up to date. I did look maybe a month or two ago at some of the 1 or 2 of the preprints that were looking at some of the viruses recovered from some of the mink farms where the mink are then culled. There were some locally here in Utah where this, I think, millions of mink in some cases are getting culled and to try to avoid any potential spillover back, to humans. Still, I think not totally clear that that's happening, but or how frequent that might be, but obviously a pretty obvious source reservoir potentially for that. And so I don't know the answer off the top of my head whether there's been enough analysis of those genome sequences to start to try to put explanation to how it is that the virus moved from humans to minks. My sense is that the virus itself is pretty promiscuous, right? It has the ability to infect several different hosts without any changes, really. However, I think what we'd predict is, and actually that raises an important point, right? Is that I think we're all kind of tuned up a little bit like geneticists, right? We have this curiosity, which is, okay, if there's a change, or as you see a behavior change, a phenotype change, like switching a host or something like that, can we put it together? Can we link that to a genetic change? The genotype has the virus recombined, or is there a specific mutation? And that's that's sort of, I think humans are kind of wired this way. Like we want that link, and we want to kind of get to the basis of it. But that can be that impulse, I think, can be a little deceiving for what we're actually seeing, because with viruses in particular, there's just so much of this, right? We were talking about this, like recombination is just happening. It's not surprising. It's diversification. Point mutations are happening, and most of them are just going to be neutral. They're not going to, there'll be a difference in the genotype, but the phenotype will be identical. And so having the tools or the ability to discern between the two, to know or to have evidence that this change mattered while this change didn't, this one did, that gets us into, I think actually in some ways why you and I launched TWiEVO was, I can't look as a genomics person. I can't just look at a sequence and say this. I need to talk to a virologist. As a virologist, you might say, well, how do I put this together as well? Or how do we think about what the changes, sampling the changes, getting that data sort to have a hypothesis to test? And so, yeah, we're still working it out. And that's why in real time, it's a great question. Yeah.
Thanks, Sandy, for your super sticker. Really appreciate it. All the donations, by the way, go towards our expenses, but also our new studio in Manhattan, which will be opening soon. And
really exciting.
We appreciate that. So here's a question which is right up Nels's alley. Could you talk about the connection of specific recombinase proteins to mobile genetic elements? Are mobile genetic element recombinases good phylogenetic markers for MGEs generally?
Oof, Connor, great question. So this is really, and maybe I'll just try to get to my punchline, which is, I don't know if I'm going to say anything useful or intelligent here, but this really does kind of point to some of the stuff we're interested in my lab, and the impact, so mobile genetic elements, let's define that a little bit, right? So, and I was kind of maybe fumbling towards this a little bit a few minutes ago in some of our conversation. So these are almost kind of virus-like entities things called retrotransposons, transposons, little kind of genomic hitchhikers almost, sequences that have the ability to over-replicate relative to sort of a regular Mendelian reproductive process. That could be, it could be an endogenous retrovirus that, so this virus gets into the host genome. If it still has that activity, as Connor's pointing out here, the recombinase proteins or the activity of the reverse transcription, the ability to copy and paste or even to cut and paste. Copy and paste is much easier to over-replicate in those scenarios. So if there are mobile genetic elements that encode those things in genomes, then you can imagine that you have sort of the machinery can be deployed to basically start to push the process forward, right? So you could imagine, like, if this is if there's an active mobile MGE, mobile genetic element, in the context of an infected cell, now this might sort of propagate diversity as well. So that's maybe the first connection. The second one, so are these good phylogenetic markers for mobile genetic elements in general? I think the answer is it depends. So in some cases, yes. So if they remain intact, so if there's natural selection on them to continue to be functional, these are the ones you see because they're the ones that over-replicate. They're somehow successful. Now, this does weigh against the fitness potentially of the host genome that's hosting that mobile genetic element. If these things are massively good at copying and pasting, of undergoing these sort of like recombination-like events. But let's say that happens and it replaces, you copy and paste, but you paste over something the host needs. That will actually weigh on the host, and you might crash out the population of, or any host that contains that will not last in the population very long. So what you end up with seeing then are, some of the relics of these proteins, and so, or the genes encoding these proteins, and depending on when they crashed out, or that activity was lost, that will the phylogenetic signal, the ability to use this to understand how related those mobile genetic elements are to host or to themselves, that starts to fade a little bit. And so I think I would say it depends. I wish this is actually where my old neighbor across the hallway here from LD Lab Studios, a fellow called Cedric Fichat, now runs his lab at Cornell. This is his wheelhouse. And so I kind of would just lean in. And before he left, I would just walk across the hallway and then pull
Ask
him
in.
Right? Yeah. That's right. Exactly. Great question.
It is. Should we jump into some of our local emails, Nels?
Yeah, let's do that. So let's, before we do that, so just to maybe just wrap on this one. So I think it's really worth it to keep an eye for recombination happening. Just as one of our listeners pointed out, as we continue to step through the pandemic, right? So, some hope that at least in some places we're starting to deploy the vaccines, but in others not so. And so there's going to be more. SARS-2 is not done. There's going to be more recombination. There's going to be more variants, et cetera. And so remembering that in addition to some of the point mutations that are sort of trotted out as the consequential ones, to remember, how there are other mechanisms here, recombination in particular. I did just want to, before we get to our local letters, I thought maybe we could just do a quick update on the variant frequencies. So we looked, I think last time we put up the link to covariants. And I was just kind of peeping at it right before we came live here.
I can put it up if you'd like. Here we go.
Oh, sweet. Yeah. And maybe pick that per country view of covariance. So this is a nice visual tool that's taking, once a week or so, some of the major variants there that are shown on the left, and then doing that frequency. So this is the same as figure 2 in the paper that we were just discussing, not just, but looking at more variants. So not just the B117 and, 5 variants, like this is more like 15 or 20 variants. And so you can see, I think, that's the UK in the top left there.
Yep. Oh, look, you can hover over it. You hover over it and you get a detailed analysis by week. Look at that.
Yeah. Isn't that neat? And amazing. So that's B117 there. that is still returning at 98% frequency or so.
Yeah.
Of luckily diminishing cases in the UK, but those are the ones that are coming up. I think I did hear that in some of the sampling recently, it's not on that graph because it's still so rare, but some of the variant that's starting to dominate potentially in India as well is starting to show up in the UK.
I'm
So
trying to find US here.
Yeah.
Where the hell is it? It's not
up at the top. You were there.
Top.
So, yep.
Oh yeah. Sorry. Here you go.
Yeah. No, you're good.
So this is what's dominating this one in here, which is
this is again that nomenclature, right? So this is now based on the 501Y.
Yeah.
And it's even more nomenclature. So it's like version 1, version 2, version 3. That's B117 basically is one way of decoding it. And so it's come up and I think in our last podcast, it was at about 46. And on that date, which would be the end of March. So last month, it was about 46% frequency from the sampling. Yeah. And if we go out to today, it's actually gone down. I think it's at, if you go to now it's 42.
42. Yeah.
And you can kind of see that little right below it, that pink
right there
wedge. Yeah. So that has increased. So the 484K has come up in frequency in the last month. That one was originally from your neighborhood in New York in those.
That's right. Yeah.
Remember the wintertime surge. And I'm wondering if that little uptick at the end there is actually what we've been seeing happening in Michigan in the last few weeks. We have now these, kind of regional flare-ups that we're beginning to see. And so really interesting, right, to see the differences here where the U.S., even though, so I think by now it was, this delayed appearance that we've been talking about over the last few months, but I don't think I would be a little bit surprised if it's the same outcome that we see in the UK, that we see in Denmark, that we see in Germany. I think that there's enough other variants here and sort of regional responses, including vaccine rollout, including, restrictions and social distancing kind of mandates or optional situations. And so, none of this is any kind of a controlled experiment. There are caveats with the sampling that we've discussed a little bit. And so you should take all of this with a grain of salt. But I think the bigger patterns, there's some interesting tidbits in here and how these viruses are circulating. Another note. Go ahead.
That's a good point because, you have to take population effects into account. We've been saying this, all along, and these variants do different things in different countries because, as we have said, they have changes in fitness, and we're really looking at the competition between the variants, not so much they're what they do in people, but whoever wins, right? And that's what goes on. So,
yeah, and that's happening, and I think it illustrates, so in one sense, in the U.S. right now, we're at this weird intermediate zone. We're kind of almost on this plateau of, I think, something like 50,000 to 70,000 cases a day being reported. And that's too much, by the way, for we're not there yet. The thing that's encouraging is the vaccine rollout. The thing is more encouraging worldwide is if we look at countries like Israel, if we look at countries like Denmark. And so in Israel,
someone asked about Israel, so I put it up. There you go.
Oh, yeah. Great question. I've been looking at this too. Unfortunately, and I don't know what's going on here, but you can see so actually that is B117 or the 501, one of the 501Ys versions. And then the data stops a few weeks back. The last data point there, I think, is Feb 22. And I don't, so why did it stop? I don't know. One guess is that the cases are getting so rare that you just don't have the amount of sampling is low enough that they're not pushing the numbers where it just doesn't get past a filter cutoff for covariants to include it. I mean, that's a great outcome in some sense, although I think there's still a few cases, but it might not be given the statistical challenges of having some signal here, the noise in this collection, you really want to get to a number. And so it might just be below a threshold as cases are diminishing. And so it kind of stopped.
Yeah. Well, here, look, there are only 70 cases 70 sequences we're talking about here on this week, right? So they've got most of the population immunized. So that's not surprising, right?
That's right. Yeah. But that's one of these ironies, right? So, this success story in many ways, and actually I was just seeing a newspaper report with folks really kind of approaching normal life because they've really driven it out with high vaccine numbers. But this is where surveillance is harder because there are fewer. It's harder to get a sample from a patient. There are fewer patients. But I think knowing what's happening will be really instructive, really important in terms of squelching this thing in some ways. I think Denmark is a similar scenario. So I think in Israel, the B117 was coming up in frequency. In Denmark, it looked a little bit more like the UK. There, the numbers are really, overall numbers are crashing as well. So in there you see it again, right? So Denmark,
they have
the
a
numbers
bit more
are higher
than Israel. They have a bit more than Israel, yeah.
But then it doesn't go all the way to the right to the, again, I think that's fallen. Yep. I think it's fallen below the threshold of having enough cases. And so with that combo of human behavior and vaccines looks promising, but let's remember that this is, whether we're talking about this for Israel, Denmark, UK, this is only a handful of very well-resourced countries. Yeah. And the question, the bigger question that's lurking is, what about the rest of the world? How are we in terms of, how do we move from, South Africa's numbers there?
Only one.
Yeah, that strain that variant has dominated.
Amazing.
In South Africa relative. There are others there. So the gray, right? These are, in this case, others, it's all other. Yeah, that's all others as well. There again, low case or sampling numbers. And then that doesn't necessarily, cases are going down, I think, in significant ways, but it also, the amount of sampling also really matters here too, and how good the surveillance is.
I mean, look at Brazil. There's no sampling really at all. There's 9 sequences.
Yeah, exactly. Right. And with all kinds of very little sampling, lots of cases here as well. I think some recent efforts to try to get the surveillance better, it's really crucial. And this, I think, also just illustrates, right, is this isn't just a public, like country by country. This is a global public health issue, ongoing health issue. And how do we get these resources distributed throughout the globe? That's really the big question, and how the sort of next chapter in SARS-2 evolution in the pandemic ahead.
I want to show a picture to our listeners here of what HIV does, and because we're doing a lot of sequencing, and we're surprised at what we're seeing, but in fact, the HIV that pandemic was the first pandemic to be sequenced extensively, and it revealed variants too. And in a recent lecture, I showed this slides, which I have here. I want to get that up here. Where is it? Here we go. This is very cool. This is let me make this bigger. Okay, so these are the subtypes that diversify from group M, the main HIV. And you can see what happened here is you had the original spillover, and then it spread through populations, first in Africa. And all the variants, they're all shown here in colors, all emerged in Africa. And then as people brought it elsewhere, whatever arrived in the country predominated. So in North America, we mainly have subtype B, right? In it went away. I don't know where it went. Oops. But
Yep. No, your point is taken. So the different colored pie charts there from country to country. It's interesting to think about, and this is just totally speculating, but the number of founder events, whether that even, so I guess are we talking about like 50 years ago, something like that, 40 years ago?
Yeah.
Versus today, and even the differences in global travel today, and possibly the difference in multiple introductions. Of course, it'll get complicated again, just different viruses, different replication cycles, and just sort of
sure. But if I remember, Nels, when the subtypes are identified, they said, well, do these have some different phenotypic properties? And the answer in the end is no. I asked Paul Benash, are these any of these strains? He said, no, they all behave the same. They're just sequence variants, and that's what happens when they move through populations. And so I think we have a little bit of that here, that we're seeing these variants. We don't know quite what to make of them.
No, it's a really good point. And again, it's a little bit of, I mean, this happens in science all the time. And very natural that we depend on our intuition, or this idea of, oh, well, we see this variant, there must be a explanation for it. And keeping a little bit in mind that, and this is a theme of evolution, right? Sort of neutral theory, that of the alternate hypothesis that does there need to be explanation, like that a genotype, a difference in phenotype, and that the genotype is linked. There are many genotypes that could lead to the same, and that's exactly what you're saying here. And Paul Benash's point there as well. Yeah.
How about this one, Nels? What about convergent evolution? Could the South African variant have arisen in different countries? Yeah. What do you think, Nels? Yeah,
that's a great question. So I think what we are seeing a type of convergent evolution in some cases, and that's even why naming the virus is sort of on the 501 mutation, the 484 mutation is starting to happen. I think in the US, there was some good evidence. This position 677, these are all amino acid positions in the spike protein, might have independently emerged by convergent evolution 7 times or something like this, even over kind of in the fall to winter, early winter surge. That's different. So a little bit different though. So when we talk about the, the variant as a lineage, in those defining mutations, so the equivalent, I'm forgetting, is the South African variant B1351 or something like that.
Yes, that's correct.
Okay. Yeah. So, similar in terms of like 20 or so, maybe a few fewer, but in that ballpark of defining mutations together that sort of hitchhike within, and then there's still more variation on top of that. But that's sort of what's defining that lineage. And so in order to hit on those like 20, it's basically like lightning striking 20 times. That's pretty, so to get a virus that would start in a different lineage and then start to show up within that lineage, not impossible, but again, like really tough. So this sort of violates parsimony, where you would actually get, so if it would be easier if every one of these mutations mattered, right? Because, so that's certainly when we think about like the single mutation that you see convergently, that's probably a mutation that does have, there's a functional consequence there. Natural selection is seeing that, something about 501, 484, 677. And again, that doesn't necessarily mean a major, massive, like game changer, look out, but enough that it's, that it comes up in frequency in the population. But then to get to that level of, the hitchhiker mutations, the ones that probably don't matter, but just happen to be with that, those defining mutations, that was really rare. And so the idea that one could kind of jump from one lineage and move to another based on that set of kind of linked mutations or hitchhiking is pretty vanishingly small. It's a great question. It's a great, and a point I was trying to make earlier, but that I think hopefully that helps crystallize it a little bit. It's an important question. All right. Well, speaking of questions, Vincent, should we move to the mailbag here?
Yeah, let's tick a few off here. Why don't you take that first one, Nels?
Sounds good. So I'm trying to remember. We have a few that we already read.
Yeah, that was my mistake. I couldn't remember, and so I guess the first one I think you have there is Tom's that we haven't done, right?
Okay, yeah, I think that's right. Let's try that. In case if we have done it, our memory is short. Probably our listeners' memory is short. Maybe Tom would be the only one who would remember whether we did it. But actually, I do think we read that one. Now that I'm reading it, it looks familiar.
You think so? Okay.
I do. So I'm going to go down the list a little bit to Chris. So Chris writes, there have been a couple of studies that looked at SARS-CoV-2 virus variants arising in patients that couldn't completely clear the infection. Could this be a method to explore how a virus is likely to evolve and escape an immune response? Maybe an immunocompromised model that mounted an insufficient immune response could be developed and then sequence the virus genomes over time to see which variants begin to dominate. Maybe this could be a way to see where an influenza strain is headed before it gets out there and cut it off at the pass. listener since 2015, starting with This Week in Microbiology, TWiM, and electric engineer from Denver, Patreon supporter. Chris, thanks for your question. And I think a really some interesting points here and some cool ideas to chew on a little bit. So, and perhaps overlapping actually with the theme of our show today, which is how in co-infection scenarios, how could recombination happen, or how frequent is this? And I think bringing in the notion of immunocompromised patient, right? So what this does is extends the window of infection, and just even in potentially like an asymptomatic scenario. But if the infection is lasting longer, then the chance of co-infection now goes up. And so you could imagine that would facilitate diversification through recombination, et cetera. And then kind of moving into the model idea. And so, this is really interesting and kind of tugs at my heartstrings as someone who grew up in genetics and thinking about model systems, model research systems. And so what would an immunocompromised model? You could imagine, so there's certainly like mouse strains, for example, right? With compromised immune systems, and whether you're doing, coronaviruses or influenza, as Chris is suggesting here, I think the trick becomes, is how good is your model to real life versus, human biology. And I think it's, in some sense, for some of the questions, like the really specific ones, to say, okay, can we do virus forecasting? Do we know which variants will arise in that model and so we kind of like do the experiment ahead of time and then try to use that information to know what might happen for us. Currently that could guide a new vaccine for influenza, seasonal flu, the next year or something, 2 years, based on what we see there. There are ferret models, for example, that are to try to get at transmission. In the end, I think the actual viruses and the actual human behavior, the actual human diversity as well. So it makes this really challenging, makes this really hard to think about effective forecasting, to think about effectively cutting it off at the pass, which would be spectacular. I love that goal. I really like the idea. I think there's a lot of practical considerations that make that really tough to pull off. But not to throw that out because I think it depends on how you define success here, right? And so I'm thinking about, you might not get to like the specific set of every mutation, but you might learn something about one or two common mutations or important mutations that sort of go beyond the specific model immunology or infection biology. That could actually give us some clues. I don't think it would maybe be enough to really say with any probability this is exactly what's going to happen in the future, but it definitely gets us a little closer. And more so the reason, in my lab and others, I think we continue to try to think about model systems of how do we model some of these events is maybe not to necessarily know what any specific virus might do or any pandemic, what might happen from year to year, but to understand a little bit of the rules of how these viruses evolve at all and what are the kind of pressure points there. And so, but it's a delicate balance of how do you get useful information with the necessary compromises you have to use when with a model system.
Nels, you remember our conversation with
Marco
Jesse Bloom, who, they changed every amino acid in the receptor binding domain. And I mean, you can do that and say, okay, these can't change, right? Yes. They knock down bindings, so they're not going to arise. So you can immediately get an idea. Although it's just hard to know what's going to happen in an infected person, right?
Yeah. Oh, but that's a really good point, is that there's it's a great example of there's information in what won't change as well, right? Under all, it's a really good point. So there are, and I was thinking back to even an earlier conversation with Marco Vignuzzi. So they've done some of this stuff with arboviruses, viruses that move from insect hosts and then to humans, things like Zika, others. And in some of those model cases, so instead of going from a mosquito to a human, they go from mosquito to mouse and back and forth. And then they actually said, oh, look, we found this mutation. And if you look at 5 years ago in a different country where Zika emerged, we see that mutation. And so there is, I don't want to pour cold water on this idea because there's really kind of tidbits that you can learn by taking this approach, but I don't know that you can get that whole, like that really satisfying, this is really how it's going to play out into the future. But that's an area of increasing resources, research. And so stay tuned, because I think there are some like great developments and real possibilities there.
I think it would be interesting to take the spike sequence and just map all the changes we have seen. And, they're not going to be a lot, frankly. These variants have pretty much similar changes going on. But you might get an idea of, and then you could compare that to Jesse's data, and anyone else has done that. I think those are useful exercises to do.
Yeah, I love that too, because it's kind of, speaking of the engineering, it's sort of the engineer's view with systems we set up, and then comparing that to sort of the natural view, and how do we learn off of each approach, each kind of complementary perspective in new ways. I think it's really cool. Yeah.
By the way, folks, if you're so inclined, please give us a thumbs up. If you go just below the video, there's a little thumbs up. You can hit that, and that kind of awakens other people to what we're doing here. Should I take the next one, Nels?
That sounds great. I think it's Raf. Yeah. Yep.
Hello, TWiEVO. I'm a listener of all Microbe TV podcasts, but have done some grad work on molecular evolution. Besides being an engineer and bioinformatics scientist, I'm also an artist. Much of my work is inspired by my technical and biological training. I wanted to share a piece of art I created titled Evolution. It depicts a progression from a molecular soup to a rather modern fish. It took a long time. It is drawn with an ink artist's pen in a fan-like notebook with 24 individual pages. I was given the book on my birthday in 2017. So Evolution is ink pen 88 by 6.25 inches. Follow the evolution from the chaotic soup on the right to the critter on the left. Let me share this with everyone so you can see this.
Yeah.
So here it is. Started October 9th, finished March 13th, a bit over 17th months. Large images of page progressions and final complete image are on my Flickr site. I include a small image of the book and complete picture below. And so look at that. This is just beautiful, right?
Yeah.
There's the soup on the left, right? And here's the final thing.
That's really neat. We'll get that Flickr. Let's get that Flickr link into our show notes so folks can go through that progression.
We'll put that in. And in fact, I will paste it in now for folks to see.
Yeah. Yeah. Please
by the way, Nels, here's an interesting comment. The best part of TWiEVO is being taught by Nels and his Neanderthal beard.
Yeah. Well, Ronnie, thanks, brother. I'm doing what I can over here. Want
to do another round, Nels? Is that good?
Another round of questions?
Yeah, there are 2 short ones coming up, right?
Yeah, let's do that for sure. Okay, so next up is Jennifer. Oh, just actually to finish off Raf's note, thanks for all your super interesting podcasts. It helps me get through the day, working from home and coding all along. Best regards, Raf. Hey, Raf, thank you. That's a really inspiring book that you've been working on. Really cool. Okay, so Jennifer writes, greetings, TWiEVO. I'm a huge fan of TWiEVO and Microbe TV. I am a registered nurse mom of 2 boys aged 11 and 9, currently homeschooling through this pandemic. We were recently researching Charles Darwin. My 11-year-old son came across some information which we thought kind of debunks the popular held belief of survival of the fittest. My son's research found that Darwin actually said that adaptability to change was what determines the survival of a life form. Obviously, we need to read all of Darwin's writings to know whether this is true. We are hoping that maybe you have some insight on this topic. Thanks for the engaging and fascinating work that you're doing and for sharing it. It is enlightening and greatly appreciated. Well, thank you, Jennifer, especially being a registered nurse, pediatric nurse in the last year or more. That is a high degree of difficulty added to the job description for sure, as we've been navigating the pandemic. Add on top of that, trying to figure out schooling for 2 boys. at home. So kudos to that you're exploring these ideas, and I'm glad you're getting some enjoyment out of some of the work we're sharing. So yeah, this idea, very famous idea from Charles Darwin, survival of the fittest, and whether how do we think about that? And so this is, in terms of adaptability, in terms of is it always survival of the fittest? And I would say the field has grappled with some of these ideas for, now decades, even longer. And overall, the idea of Darwinian evolution, broadly speaking, still holds pretty well, holds up over hundreds of years in terms of some of the just kind of main ideas. But like I think any sort of successful thoughtful theory or thoughtful approach, survival of the fittest can be kind of deceiving in some cases. There's also actually some really interesting ideas or a takeoff on survival of the fittest called survival of the flattest. And the idea there, so pioneered by Paul Turner, who was on TWiEVO. I think we caught him maybe at the ASM annual meeting in person.
That's right.
Yeah, he's doing some really cool work on viruses. In this case, viruses that infect bacteria and lyse them, kill them as a possible new antibiotic. Some incredible work. But before that, some really a little more foundational work, I would say, on the idea of in populations, some things like resilience of the population, the being the flattest, not necessarily the fittest. Can you persist? And part of that, so part of that thinking is, mostly how we as humans interpret our own language, I would say. So what do we mean when we say fittest? And how do we define that? And I think so defining it in a very strict, limited competition, I'm a little better, or I took this thing and kept it away from that thing. That view of fittest is limited. And so there is absolutely, it falls short in understanding sort of the evolutionary process. So, when we talk about symbiosis, organisms sort of seemingly teaming up, cooperating in some ways, that starts to challenge, I think, some of the simple notions of fittest and what that means. How could like a cooperating or compromising almost biologically doesn't necessarily fit that very simple view. So in either case, the idea of adaptability to change determines survival of a life form. So so adapting is kind of the sort of the work of evolution in a sense, right? Is that if an environment changes or an animal moves to a new environment that has different features, or if maybe that your environment changes because it was invaded by a population of viruses that your immune system has never seen before, then we do see as outcomes of evolution the adaptive process. That could fit into, the kind of simple idea of the fittest, but it gets more complicated. It's worth pursuing and worth kind of continuing to chew on and learn about. So yeah, I guess that's kind of a long-winded answer that probably isn't super satisfying, but but that's what I got at the moment. Yeah.
All right. One more from Fernando. Hi, Nels and Vincent. I sent the message below to TWiEVO, but given their heavy mailbag, it's likely queued for sometime in 2022. Nevertheless, I feel it's relevant to the discussion of SARS-CoV-2 recombination in TWiEVO, which I just finished listening to on my Saturday morning walk. The weather on the North Sonoma coast in California is gray, overcast after overnight rain, light wind from the west, 56F. In brief, a Portuguese teenager without known pre-existing conditions had 2 COVID-19 episodes with 2 genomic variants, and the genomic evidence suggests they coexisted for some time. And he gives a link to a medRxiv preprint. Thank you for shining the critical light of evolution on the latest and sometimes panicky COVID-19 news. So this is basic. This is totally relevant to this episode. And what Fernando had sent to TWiEVO is this preprint. 17-year-old, very long COVID-19 episode with 2 peaks, and he had 2 different variants concurrently. And so Fernando asked, could such double infections enable recombinations? that accelerate the emergence of multi-mutation variants like the infamous B117. And I think you just listen to this episode, right, Nels?
That's right. So actually that was, and we were, I think, ruminating on that a little bit. So this is great because it helps to give a little bit of framework here, a perspective. So I was speculating, I think, or maybe we both were, that B117, given some of the, and this was back, I think, like December or January, that given, we were looking at some of the early reports, and just like Fernando is pointing out, sort of this flurry of mutations all seemingly showing up at once. Right. Going back now and looking at a little bit more data. So the way we were presenting it, I think we even put it up on the screen, was that so it was like time on the x-axis and mutation number on the y-axis. And then we were looking at most sort of some sampling of coronaviruses, SARS-2, and closely related lineages. And then all of a sudden B117 sort of popped up above the curve with sort of variants from the original SARS-2 isolated, I think, late 2019, one of the first genomes characterized, and it sort of like jumped. If you look though or add more sampling of other SARS-2 in that lineage, you can almost see like the mutational pathway of accumulation where you don't necessarily invoke recombination. Recombination is still probably happening, but maybe not like bringing the 10 defining mutations with the other 10. And so this, the work we just talked about today, is B117 already exists, right, on the timing of this, and then it's having enough differentiation to say it itself is recombining in some new ways. And so, yeah, but that doesn't exclude that. I wouldn't be surprised if there was some recombination happening leading up to B117. It's almost just with the frequency of this, it's probably almost a foregone conclusion, but probably not one kind of like haymaker event where all of a sudden it just leaps off the curve somehow and grabs all of this other variation in one go. Yeah. But yeah, I agree. This is great, Fernando. And exactly more evidence for co-infection, which I think is starting to, and this is perfect kind of illustration of how science, progresses, is you might have one hypothesis with some evidence, but then there's a complementary piece of evidence and another, and it is kind of, that's really what sort of slowly advances it and starts to give better resolution to our understanding of the biology here. And I think this is very consistent with the, with what we've been kicking around today.
All right. Let's
Sweet.
do some picks, Nels.
Yeah, I'm excited from this pick, Vincent. I know, did you see this? The Mars rover helicopter called Ingenuity just pulled off its first successful test flight. Oh my goodness.
Very cool.
So fun. So this, I think it's like a 4-foot rotor span on the Mars helicopter. We'll put this link up from NASA. And they've been kind of deliberately getting this going. So dropped the helicopter, I think a couple weeks ago, even from the main rover that they just launched up there, successfully landed up there and is running. And then the main rover backed away and you just saw this little helicopter. I think it even has a little flap from the wing of one of the original Wright brothers airplanes that's been sort of patched onto it. And so they've really been branding this as a Wright brothers moment. I think with some, with some of that really cool pioneering advancement, I think it fits. I think they even named that location on Mars Wright Brothers Field or something after the successful event. So anyway, yesterday, I think it was, we're seeing now they're firing up the 2 rotors of the helicopter. It comes up, hovers, I think, a few meters off the Mars surface for 30 seconds and then touches down successfully as sort of and that's really cool. There's something to see it then recorded, a little video from the Mars rover itself. Wow. What fun. Cool. What an amazing time. Yeah.
Science, man. Good science, right?
not too shabby. How about you, Vincent? What's your science pick of the week?
So I want to draw people's attention to the latest episode of Twin This Week in Neuroscience, another podcast that I do, number 17, which I just posted before we started. It's just audio, but it's with Michael Niederbach, who's a professor at Yale, and he studies C. elegans, and he and his colleagues, collaboration with a postdoc in Horowitz lab at MIT. So worms, like C. elegans, they don't have eyes, but they found they respond to light. And in particular, they respond to bacteria. So the worms eat bacteria in the soil.
Mhm.
They like to eat E. coli, but now and then they'll encounter a Pseudomonas, which makes a toxin, and they don't like that. So they actually can sense the light. It's a blue toxin. They can actually sense that and move away. And they identified the genes that are involved. It's an amazing piece of work. So they have this wonderful assay for the worms moving away from a light source. And then they made mutants. And they found that a couple of stress proteins are involved. And they're not light receptors at all. So how this works is just going to be interesting to sort out. But Michael is great. He's really good at explaining. He's got a great view of things. So I would check it out, listen to it. There's no video because the video didn't work out this time, but can listen to it.
I'm going to, on my menu, definitely check it out. I've followed just from a distance Mike Nittebach on Twitter. He's a pretty interesting, colorful guy with some pretty unique ideas and interests. So I would recommend that too. Tune in, and also maybe if you're Twitter addicted, like some of us, check that out as well. Some really interesting stuff.
All right, that'll do it for TWiEVO 67. You can find the show notes at microbe.tv/TWiEVO. If you want to send us a question or a comment, TWiEVO at microbe.tv. Although, Nels, as we do more and more of these live streams, maybe the email becomes, like archaic. I don't know.
Vestigial organ, like the whale's legs. Yeah, we're letting go of our...
But the chat is limited to some characters, so you can't get into these long, which I like. So I think we'll always have it. Anyway, that's that's TWiEVO at microbe.tv. If you like what we do, we appreciate your donations tonight by the super chat and super stickers. But if you'd like to do something else, microbe.tv/contribute. Nels Eldy is at cellvolution.org on Twitter. Earlybird. Thank you, Nels.
Hey, thank you, Vincent. And also a big thank you to our listeners today coming in again for the live broadcast and those super stickers, those donations in real time. What a shot in the arm for us. That's really cool. Thanks, everyone. Really appreciate it.
Oh, Sean just gave us a super chat. Thank you so much, Sean. Simon asks, when's the next live stream? Nels and I will have to, it's going to be in about a month. We do these roughly every month. And I think Wednesdays at 2 p.m. Is probably the good time. So I'll let you know. I'm Vincent Racaniello. Yes, go ahead, Nels. What were you going to say?
Oh, I'll also try to get that up to tweet that out too, just to get a little heads up. But yeah, that's a good rough time for when we'll be back.
I'm Vincent Racaniello. You can find me at virology.blog. Music on TWiEVO is performed by Trampled by Turtles. You can find them at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.